this lecture is on the structure of the respiratory tract. The objectives of the lecture, at the end of this session you should be able to describe how the respiratory tract is designed to perform its functions at different levels, and also you should be able to identify and describe and also to illustrate, that means to draw a small simple diagram of a microscopic section of the trachea, the bronchus, the bronchioles and the alveoli. Okay, so with these objectives in mind, we'll start. Now if you look at the component parts of the respiratory tract, we generally tend to divide it into two parts, that is the upper respiratory tract or the upper airways, which consists of the nose, where you have the mucocutaneous junction. If you examine your own nose, you should be able to see that. You can see the skin coming in, continuing as the nasal mucosa, and then that will open into the nasal passage. Okay, and then the nasal passage is connected to the pharynx, which is connected to the larynx, and then the trachea. So air takes this pathway when it goes into the lungs. Now after the trachea, you come to the lower respiratory tract, which consists of the bronchi, the bronchioles and the alveoli. Now we will learn a little bit about the structure of each of these different parts. Now before really going into this, because we are always talking about how the structure is designed for the function, we'll take a small brief look at the functions. Now as you all know, the respiratory system is designed to take in air, inhale air, and then to expel air. And when the air is inside the lung, there will be exchange of gases between the air and the blood. Okay, so when the before the air goes into the lungs, the air has to pass through the upper airways, and the upper airways, we say, are involved in what we call air conditioning. That is taking the outside air and conditioning it so that it is not harmful to the lower airways. What the lower airways does is to allow gas exchange. The bronchioles and the alveoli are designed for this exchange. And then finally, the air which is taken into the lung has to be expelled to the outside environment. Okay, now we will learn about the upper respiratory tract. As I told you just now, we have the nose and the nasal passage. Now if you carefully look at your nostrils, examine them, you can see that the first few centimeters of the nose has small hairs in it. Now what these hairs does is that they trap any kind of large dust particles that goes into the nose. I'm sure you have felt this. When a dust enters the nose, you get the small particles getting trapped within these hairs, and sometimes you will sneeze and get rid of those dust particles. Then of course the nasal passage is lined by the what we call the respiratory epithelium. Okay, so the respiratory epithelium, what it does is it humidifies the air, which is quite dry. Sometimes the air that we breathe in can be dry, so it's necessary the respiratory epithelium will humidify the air. Humidify means it will add water to the air. So how this is done is because there's a lot of mucus glands in the respiratory epithelium, and the mucus will help to humidify the air. Then the nasal passage is also richly supplied with blood vessels. So there's a lot of blood coming to the nasal passage, and this helps to increase the temperature of the nasal passage, and thereby the inhaled air which gets into the nasal passage also gets heated up to the body temperature, because before it goes into the lower respiratory tracts. Okay, then of course we have the larynx, The larynx are specially designed actually to produce the voice and also to prevent from any large materials from going and blocking the respiratory passage, because if the respiratory passage gets blocked, then there won't be any oxygen to the cells and the person will eventually die. So because of this, the larynx protects the lower respiratory tract without allowing any materials to get in, and the epiglottis, as you know, will close off the larynx and prevent any swallowed materials from going into the respiratory tract. And also larynx are extremely important in voice production, which you will be learning later. Okay, so let us look at the structure of the respiratory epithelium. Now you can see this H&E slide of the respiratory epithelium, a very high magnification, and here you can see a pseudo-stratified columnar ciliated epithelium with goblet cells. Okay, so it's pseudo-stratified columnar. You can see that there are several nuclear layers. However, these all belong to cells which lie on the basement membrane. Though it appears as if there are several cell layers, this is actually only single cell layer where the cells are of different sizes, and because of that it looks stratified, so we call it pseudo-stratified. And on the luminal surface you can see the cilia. As you know, cilia are visible in light microscopy and you can clearly see the cilia. Now the cells which the goblet cells don't have cilia, the other cells will have cilia, and you can see the goblet cells, the nice goblet shape of the cells, and the cytoplasm is full of mucin. Now the respiratory epithelium, we call it the respiratory epithelium because it lines the nasal passage, the nasopharynx, the larynx, the trachea, and the primary bronchi. Now the pseudostratified epithelium is quite a thick epithelium and it protects the underlying tissues. Here again you can see even higher magnification where you can observe the ciliated cells. This has been outlined. You can see the nucleus and the cilia on the luminal surface, and the cell is lying on the basement membrane. All these cells are lying on the basement membrane below. And then you see the mucus producing cell which has a lot of mucin inside it, and it has a goblet shape. Okay, so we call this the mucociliary escalator. Escalator is, it moves only in one direction. The cilia transport the mucus in one direction. Here again there's another picture for you. Here you can see the cilia, the different nuclei, the goblet cell, right? And you can visualize the pseudostratification and cilia and the goblet cells. Now what is the mucociliary escalator? As I told you, the goblet cells produce mucus which is secreted onto the surface of the epithelium. Then the cilia on the ciliated cells, they beat like a wave-like pattern towards the upper airways. Okay, so the cilia always beat towards the upper airways, and because of that, because of this wave-like action of the cilia, the mucus, the layer of mucus on top moves in the direction of the upper airways. So all the time the mucus is carried away from the lower air passages. So if there's any kind of foreign body, dust particles or any foreign body, it will get trapped within the mucosa, within the mucus layer, and then get swept towards the upper airways. So this mucociliary escalator is very important in preventing foreign bodies or dust particles from getting into the lower respiratory tract. Now here you can see the nasal passage with the respiratory epithelium lining it, the goblet cells and the pseudostratified epithelium, and the cilia very clearly visualized here. Okay, now we will talk about the bronchial tree, the structure of the bronchial tree. So the bronchial tree basically starts in the larynx and then continues as the trachea. Now the trachea comes and divides into two parts at what you call the carina, into the two main bronchi, the left and the right, and then these bronchus again divides into smaller parts, and you can see very small bronchi dividing into different small parts. This is a diagram of the bronchial tree. On top you can see the trachea. Okay, it comes down into the thorax and then divides into two main bronchi which go into the right lung and the left lung. Okay, on the right side you can see the main bronchus then goes and divides into three lobar bronchi. Okay, three lobar bronchi, because the right side has three lobes, and the left one divides into two lobar bronchi. Okay, and then each of the lobar bronchi again divide into what we call the segmental bronchi, which go into the different segments which are there in the different lobes. Okay, so in the right side there are three lobes, and inside those lobes you get the segments. So you learned the anatomy of the lungs, and I'm sure you have understood what the bronchi are, the lobes and the segments are. Okay, so for those you get the main bronchus to each lungs, the lobar bronchi to each lobe, and the segmental bronchi to each segment, right? And then the segmental bronchi again divide into two, right, becoming smaller and smaller until they become the bronchioles. Okay, bronchioles. And then the bronchioles further divide and their walls become thinner and thinner, and they form what are called the alveolar ducts. Okay, alveolar ducts, and the alveolar ducts only go and end in the alveoli. Alright, so starting from the trachea down to the alveoli, that is what we call the bronchial tree. So what they say is there are 23 generations from the trachea to the alveoli. That means the trachea divides 23 times before it comes to form the alveoli. Okay, so as I told you initially, the function of the respiratory system is for actually respiration, right? That is to intake the air, then change gases, and then expel the air. All right, so when we think about the function, we generally divide the bronchial tree into two systems, which we call the conducting system and the gas exchange system. Okay, so most of the respiratory tract that you can see, the gross thing, are generally involved in conducting the air into the lungs. Okay, so that is the conductive system. So the conductive system consists of the trachea, primary or main bronchi, right and left lungs, going to the right and left lungs, then the secondary bronchi or the lobar bronchi going into each lobe, then the tertiary bronchi or the segmental bronchi going into each of the segments. Okay, then the bronchioles, and the bronchioles become thinner and thinner and become what we call the terminal bronchioles. Okay, so after the terminal bronchioles they become even more thin and branches out more, and we call them the respiratory bronchioles, right? Then the alveolar ducts, then the alveolar sacs. Okay, so here the whole of the respiratory tract is divided into the conducting and gas exchange, and where you draw the line is at the respiratory bronchioles, right? So everything, all the whole of the pathway above the respiratory bronchioles does only conduction of air. It brings the air from the outside into the lungs, right? And gas exchange only happens from the level of the respiratory bronchioles, right? From the level of the respiratory bronchioles, because the other bronchioles are too thick, their walls are too thick for any kind of gas exchange to happen, right? So the a point where this changes into gas exchange is the respiratory bronchiole, which has a thinner, much thinner wall. Okay, so the alveolar ducts and the alveolar sacs are also involved in gas exchange. So if you take the gas exchange, actually most of the gas exchange occurs at the alveoli, right, in the alveoli. Okay, so we'll talk about the different parts of the bronchial tree now. So first of all, we'll talk about the structure of the trachea, right? So trachea basically starts at the end of the larynx. So you know that the larynx, if you touch your anterior aspect of the neck, just below the where the jaw extends onto the neck, you can feel your trachea. It's made out of hyaline cartilage. You can compress it a little, but the shape won't change because it's made out of hyaline cartilage, which is a semi-rigid structure, right? So the trachea also continues because that also has rings of a hyaline cartilage, right? So the trachea starts at the end of the larynx, which we call the cricoid cartilage, right? Cricoid cartilage is the ring of cartilage. That is the only complete cartilage ring that you will see in the bronchial tree, the cricoid cartilage, right? So the trachea begins at the cricoid cartilage and then it ends at the carina, where it divides into two, right? So the diagram, you can see I have circled it in red, starting from the cricoid cartilage up to the carina. So that is the extent of the trachea. Okay, so the trachea, we call it semi-rigid tube arising from the inferior aspect of the larynx. Now it's important for the trachea to always be open, okay, because if the trachea closes, no air is going to get into the lungs, right? So anywhere there is an obstruction in the trachea, then the person will die quickly without any oxygen, right? So it is important for the trachea to be open all the time, right? So as I just told you, the trachea has to maintain its shape. It cannot collapse, okay? So it can permit a little bit of expansion and a little bit of passive recoil, where it keeps its shape and does not collapse, right? That is the most important thing. Whether it expands a little is really not that important, but and also the recoil, but you must remember that the trachea does not collapse. If it does, the person will die. Okay, so here you can see a low magnification of the trachea. Actually on either side you can see thyroid tissue also, right? So this is a section through the neck where you can see the whole of the trachea cut in a circle, right? A cross section through the trachea, very low magnification, and it shows you the cartilage of the trachea, which I said is hyaline cartilage. So it is shaped like a C, right? A C, and at the corners of the C are connected by the smooth muscle, the trachealis muscle, right? So this is a low power view where you can see get a general idea about the structure of the trachea. Okay, so this is again a similar one. This is the picture from your Wheater's histology. Okay, so you can see the cartilage, C-shaped cartilage. You can see the mucosa, folded mucosa, and you can see the smooth muscle layer, the trachealis muscle joining the two ends of the cartilages. Okay, so this is actually a high power view of the cross section of the wall of the trachea. Okay, so you can see the different tissue layers that form the wall of the trachea. So the luminal side, you have the epithelium. Okay, so the respiratory epithelium will line the trachea, right? So you get the pseudostratified columnar ciliated epithelium which is lining it, and you can actually see the features in this picture. You can see the levels of nuclei, and you can very you can make out the cilia on the luminal surface of this epithelium, right? So the lumen of the trachea is lined by the respiratory epithelium. Okay, so below that you get what you call the lamina propria, which you know has connective tissue and blood vessels and lymphatics, right? That is the connective tissue layer, the first connective tissue layer. And below the lamina propria you get another connective tissue layer which we call the submucosa. Okay, so the mucosa is the respiratory epithelium and the lamina propria together. Okay, so the epithelium and the lamina propria together is called the mucosa. Okay, so below the mucosa the connective tissue is given another name. We call it the submucosa. Okay, the submucosa. So the connective tissue, the arrangement may be slightly different from the lamina propria to the submucosa. Okay, and in the trachea what is important to remember is that in the submucosa you get these glands. Okay, so submucosa you can see the glands there which secrete mucus, and from small ducts they are taken to the luminal surface. Okay, so submucous glands are there, they take the mucus to the luminal surface, right? So below that you see the tracheal cartilage, which is a typical hyaline cartilage. So you can see the chondrocytes and you can see the amorphous matrix, right? So you did cartilage when you did a histology of the skeletal system, right? So you know, so this is a hyaline cartilage where you can see the cells and the amorphous matrix. Okay, so just what I just told you, I have put it into writing. So epithelium, pseudostratified columnar ciliated epithelium with goblet cells. Okay, there are goblet cells, and this forms the mucociliary escalator which we spoke about, right? And then below that you get the lamina propria and then the submucosa. So in the submucosa you get seromucous glands. Again they produce mucus, right? And then the C-shaped hyaline cartilage which prevents collapse. Okay, which prevents collapse, and also the trachealis muscle which is made out of smooth muscle, right? So these are the typical features of the trachea: the respiratory epithelium with cilia and goblet cells, the seromucous glands in the submucosa, and the the C-shaped hyaline cartilage and the trachealis muscle. Okay, so how do you actually distinguish cross section or a histological section to know whether this is actually the trachea or not? Okay, so when you take histological appearance, actually the trachea and the main bronchi will have the same appearance, because when you come to the main bronchi also you have a large chunk of hyaline cartilage and some smooth muscle, and the same epithelium is there with the different layers, right? So a high power view of, without you being able to see that the cartilage is C-shaped, you can't actually say whether it's the trachea or the main bronchi. So both will have similar appearance in high power view. Okay, in low power view only you will be able to see the C-shaped hyaline cartilage which is continuous, right? The cartilage is continuous, there are no breakages within the cartilage plate, and the trachealis muscle is also continuous, right? So you have the C-shaped cartilage which is continuous, and the two ends of these cartilages being connected by a continuous layer of smooth muscle. Okay, so both the cartilage and the trachealis muscle is continuous in the trachea, right? But these features you will be able to see only in a low power view, right? So if you are given a slide of the one that we saw initially like that, so you can actually, whether you call it the trachea or the main bronchi, you will get full marks, right? Unless of course it's a very low power view which shows you the C shape and the trachealis muscle, then of course you will have to identify it as the trachea, right? Okay, then we from the trachea we talk about the bronchi. As I said, the two main bronchi are there. Alright, we call them the primary bronchi, and then we have the lobar bronchi, which are the secondary bronchi. Now, lobar bronchi also may be outside the lung sometimes or inside the lung tissue, but the segmental bronchi will definitely be inside the lung tissue. Alright, so these are called tertiary bronchi. Okay, so several names are given to the same structure. So segmental bronchi are called tertiary bronchi because that is a third division. Okay, and these are intrapulmonary bronchi, right? So the segmental bronchi are inside the lung tissue, so they are called intrapulmonary bronchi. Okay, so if you see a cross section of a segmental bronchi, surrounding that you will see lung tissues. Okay, now here this picture has been put there so that you will look at the cartilage arrangements, right? So in the trachea you get C-shaped cartilages. Okay, and then when you come to the bronchi, there will be cartilage rings. Though we say rings, they are not complete rings. Okay, so they may, even in cross section they will look similar. So they are not, they may be broken into chunks in the primary bronchi. Then when you come into the lobar bronchi, again you see cartilage plates. Okay, so cartilage plates will be larger in the larger bronchi and become smaller and smaller as you go down. So in this diagram you can see the cartilage plates becoming smaller and smaller, right? So cartilage will continue from the trachea up to the segmental bronchi. You will get cartilage plates. Okay, so always remember, if you see any hyaline cartilage in a cross section of a bronchus, you know that of a lung tissue section, you know that it is a bronchus, right? It is a bronchus because when you come to the bronchioles, there is no cartilage. All right, so if there is a cartilage, then you know it's either a segmental, most probably a segmental bronchus. So if you can see lung tissue surrounding it and you can see a cross section with the cartilage plate, then you know that it's a segmental bronchus. Okay, so you must remember that beyond the bronchioles there is no cartilage. All right, beyond the bronchioles there is no cartilage. There is smooth muscle and the mucosa is there. All right, and then as it goes down, the thickness of the wall becomes thinner and thinner. All right. Okay, so then as I just said, if you talk about the bronchi, they have cartilage plates, or what we call cartilage islands. When you do a cross section, you see them as cartilage islands. Then you will have a discontinuous layer of smooth muscle, and of course the respiratory epithelium, pseudostratified columnar ciliated epithelium with goblet cells. Okay, so the primary bronchi and the lobar bronchi will have this picture.
Okay, so in bronchi you will see the primary and the secondary. That means the primary or the initial bronchus, the main bronchus, and the secondary bronchus, which we call the lobar bronchus, will have a similar histological appearance to the trachea, right? So you will have cartilage plates and you will have the respiratory epithelium lining it, right? However, as you come towards the tertiary or the segmental or intrapulmonary bronchi, the segmental ones, which are smaller in diameter, you will see that the epithelium is becoming shorter, right? So the pseudostratification will reduce little by little, and you will see a simple columnar epithelium with cilia, right? With cilia, however, with few goblet cells. The goblet cell number is going to reduce. So the maximum number of goblet cells are seen in the trachea with the pseudostratified epithelium, and as it comes down towards the segmental bronchus, the epithelium becomes thinner and turns into a simple columnar ciliated epithelium, and the number of goblet cells reduce.
Alright, so here you have a cross section of the intrapulmonary bronchus. You can see the hyaline cartilage plates. Okay, so you can see you can just make out the mucosa there, right? Smooth muscle is not very clearly seen here, but surrounding it you can see the lung tissues, right? The alveolar cross sections of the alveoli are visible. So you know that this is an intrapulmonary bronchus, a segmental bronchus, or a tertiary bronchus, right? So all these three names are given to the same structure: segmental bronchus, tertiary bronchus, or intrapulmonary bronchus. So generally intrapulmonary bronchus is a term that you generally see in histology, right? So histological books we call it that because you can see the lung tissue surrounding it. Okay, so here again a very low power view of some intrapulmonary bronchi, right? So you can see the cartilage, you can see the mucosa, right, and several cut sections of intrapulmonary bronchi. Okay, so then from the bronchi you come to the bronchioles, right? Bronchioles. So initially you did your arterial system, so you started from the aorta, which has a very thick wall, and then ended up with the capillaries, which has only one layer of cells, one endothelial, only one layer of cells, right? So the lung also is similar, cartilage, the trachea with a thick wall, cartilage, and a thick respiratory epithelium, and then the wall becomes thinner and thinner and thinner until you come to the alveoli, which have only one cell layer, right? One cell layer, one simple squamous cell layer, right? So from the epithelium also becomes thinner and thinner, starting from the pseudostratified columnar ciliated and ending up with the simple squamous epithelium. Okay, so bronchioles are small diameter, so they are coming as branches from the segmental bronchi. Okay, so they have airways less than 1 millimeter in diameter. So similar to arterioles, bronchioles are generally the diameter is less than 1 millimeter, so they are very small. Okay, so the bronchioles are the ones that maintain resistance to airflow. Now remember that when you're breathing in, if the lung just keeps on enlarging, then there is no control, right? So the control or the resistance to airflow is maintained by the bronchioles. So bronchioles can completely shut down any air flowing into a certain area of the lung, right? So if bronchioles constrict, then air won't flow through into the alveoli. Okay, so bronchioles, when you look at a cross section, you will see that they have a ring of prominent smooth muscle. Okay, there is no cartilage, there is there is a ring of smooth muscle. Okay, very similar to a bronchioles, the arteriole, and there is the epithelium is also thinner. It's thinner than the respiratory epithelium. It has now become simple columnar ciliated epithelium. However, they have goblet cells, right? So pseudostratified columnar epithelium will be thicker than a simple columnar epithelium, right? So the epithelium has become thinner. It has come down to simple columnar ciliated, right? Cilia are still there, goblet cells are still there. Okay, so that is what you will see in a bronchiole. Alright, so this is a cross section probably of a slightly larger bronchiole, because you can see the smooth muscle doesn't actually go right around, because that is because of the cross section, right? In general it has to go right around. Now here you know that this is a bronchiole because it does not have cartilage plates, right? And there are cilia, though some areas of it looks pseudostratified, most of it is simple columnar, right? So always remember, because from the pseudostratified epithelium it becomes thinner and thinner gradually and then becomes simple columnar, right? So cross section sometimes may have sorry, pseudostratified as well as columnar epithelium in it. But if it does not have smooth muscle, then you know that we have to call it a bronchiole. Okay, so again, bronchioles also, there are several types. We call them the terminal bronchioles and the respiratory bronchioles. Okay, so the terminal bronchioles is the last segment of the conducting system, as we discussed earlier. Then the terminal bronchioles will divide into several respiratory bronchioles, right, which have a very thin wall and can is involved in gas exchange, and that is why we call it the first segment of gas exchange. Okay, so those are the two types of bronchioles, terminal and respiratory. Okay, so here you can see a terminal bronchiole. You have a ring of smooth muscle and a simple columnar epithelium with cilia on it, right? So as you go down in this terminal bronchioles becoming respiratory bronchioles, the epithelium will thin down more, and you find less and less goblet cells as you go down, and less ciliated cells. Okay, right? Then we come to the respiratory bronchioles. The walls are very thin. Okay, so here sorry, you can see a respiratory bronchiole, and from the wall of the respiratory bronchiole you will get alveoli coming, right? So and then the respiratory bronchiole will again divide to form what are called alveolar ducts. Okay, alveolar ducts which will open into the alveoli. Okay, so this is slightly higher power view, where you can see, because the lung tissue will just basically look like empty spaces, right, with lines. Okay, so the lines are actually the alveolar sacs which are lined by simple squamous epithelium, right? It's very thin epithelium, that's why it looks very sort of empty like, right, because it's all full of, those are sacs full of air. Alright, so here this is a electron micrograph, right? So you see the bronchiole, then you see the bronchioles dividing into respiratory bronchioles, and then the respiratory bronchioles dividing into alveolar ducts, and finally ending in alveoli. Okay, so arteries, pulmonary arteries are also there. So you get a large number of blood vessels coming into the lung, because the gas exchange has to occur between the alveoli and the capillaries, right? So you get the pulmonary artery branching out, similar to the branching of the bronchial tree. Okay, so now we'll look at a special type of cell which is found in the epithelium. Now these are called Clara cells. The Clara cells are found in the epithelium of the respiratory and the terminal bronchioles. These Clara cells are non-ciliated. They don't have cilia on their luminal surface, and in their cytoplasm you see a lot of vesicles, right? So that is how you identify them from the other cells of the epithelium. Now Clara cells are important because they secrete a component of what we call surfactant, right? So surfactant is an oily sort of substance which reduces the surface tension of alveoli. Okay, because alveoli have air inside them, and there's a pressure gradient inside the lung. Sometimes you can get alveoli collapsing, right, and getting stuck together, the walls getting stuck together. To prevent this, you have what is called surfactant. Alright, so you can read up on about surfactants a little bit more in one of your books. Now the Clara cells also have the ability to divide, right? They can divide, and also they can differentiate to replace damaged cells. Okay, so Clara cells will divide and then replace the cells of the respiratory epithelium that may be damaged, and also they can detoxify substances. They can absorb some of the toxic substances and then break them down into non-toxic components. So Clara cells are important cells in the epithelium. This is a histological slide of the Clara cells. You can see that they are found in the terminal and the respiratory bronchioles, and they have a dome shaped in their profile of the membrane, and the luminal surface does not have any cilia. Okay, so from that you can distinguish them as Clara cells. However, when you are looking at a slide, it's a little bit difficult to find these Clara cells unless you look for them specifically, because there are not a lot of them, just a few of them are there. Okay, here this is a electron micrograph, and that also you can see the Clara cells nicely. They don't have any cilia, right, and they have a domed luminal surface. So in the bronchioles, the respiratory and terminal bronchiolar wall, you will be able to find these cells. Okay, now we talk about the alveoli. Now alveoli are the end points. The respiratory tract goes and ends. The bronchial tree divides, becomes smaller and smaller, and finally ends in alveoli. So alveoli are completely there for gas exchange, and they are the sac-like appearance is there to increase the surface area. Okay, so to maximize the gas exchange, these alveoli have a single cell thick wall. Okay, so the epithelium is simple squamous epithelium, one layer of squamous cells or flattened cells, and there will be a very fine elastic and collagen fiber supporting network just to support the single layer of cells. Okay, and then the next thing we need for gas exchange is the capillary network. So surrounding the alveoli you get the capillary network. Okay, so the capillary also has simple squamous epithelium and a little bit of connective tissue, right? So the gas has to go through only those two layers of cells. Okay, so here you can see a diagram, a 3D diagram, where you can see the alveoli, which are basically air-filled sacs, and surrounding that the capillary beds. So the whole of the alveoli is completely surrounded by capillaries. Okay, so this is again a cross section of an alveolus. Okay, so we see this, you see the alveolar duct, which are branches of the respiratory bronchioles, and then the alveolar duct comes and you get several alveoli together opening into that particular alveolar duct, right? So these several alveoli which open into one single alveolar duct is called a alveolar sac. Okay, so this semicircular area, which is a sac-like thing, is called a alveolus, and several alveoli, alveolus is the singular, so alveolus is the singular, alveoli is the plural. So several alveoli, all of which open into alveolar duct, is called the alveolar sac. Okay, so if you look at a cross section of the lung, this is what you will see, these empty spaces where the air is, and a thin single layer cell wall, right? So air filled sacs, and you can identify alveolar duct because several alveoli open into it surrounding that, and then you can see each of the alveoli. Okay, and also you will see cross sections of capillaries. Cross sections of capillaries. So if you see this view, then you know that it is the lung parenchyma or the lung tissue as we call it. Okay, so this is the H&E section. This is a low power view. Right, so then what are the cells that you find in the alveolus? Now we said it's a simple squamous epithelium, but then there are two different types of cells in the epithelium or the wall of the alveolus. We call them type 1 pneumocyte and type 2 pneumocyte. Two different types of cells. Okay, so if you take a very high power view of the wall of the alveolus, you can identify the two different types of cells. So alveolar type 1 cell is the typical squamous cell that we see with a flattened nucleus and small amounts of cytoplasm spreading around, the typical squamous appearance. Okay. However, the alveolar pneumocyte or pneumocyte type 2 is slightly different. It has a larger nucleus which bulges into the lumen, right? So you can see type 2 pneumocytes at the junctions of alveoli, right? And also in a very high power view like this, you can see capillaries, cross sections of capillaries with red blood cells inside it. Okay. Right, so we'll learn a little bit of detail about the different types. Though the type 1 pneumocyte, as I said, it's a flattened cell with a densely staining flat nucleus, and this pneumocyte is completely, its function is for gas exchange, and that is why it is so thin. So type 1 pneumocytes are the cells which are involved in gas exchange. Then type 2 pneumocytes, these are rounded cells as you saw. They have large nuclei and a prominent nucleoli as well, and they are found at the junction of two alveoli, the two junctions of two alveoli, and they have a special function of secreting surfactant. Now we learned earlier that Clara cells also secrete a component of surfactant. So the other components are secreted by type 2 pneumocytes. And similar to Clara cells, they also have the ability to divide. So they may be able to help when lung tissue is damaged. When type 1 pneumocytes are damaged, type 2 pneumocytes may be able to divide and replace those cells so that gas exchange can happen again. Right? So in destruction of lung tissue, it's very difficult to bring the cells back to the normal. So these pneumocytes type 2 and the Clara cells may be actually helping to heal such injuries. Okay, so a little bit more about surfactant. Now it is a lipoprotein complex, right? It is an oily substance. It is made out of lipid and proteins, and as I said, it's secreted by type 2 pneumocytes and Clara cells. And the reason why we have this is to reduce the surface tension of alveoli. Okay, and this will prevent alveoli collapsing on themselves when there is a negative pressure. Okay, so alveolar walls, if they collapse and stick together, then the intake of breath becomes difficult, and also the inhaled air can get stuck inside or trapped inside the lung. Okay, so surfactant basically prevents this from happening and keeps the alveoli open, reducing the surface tension. Okay, so then what is called the alveolar septum. This is actually the barrier between the blood and the alveolar air, the alveolar septum. So what forms the alveolar septum? You have the type 1 pneumocyte, okay, on the side of the alveolus. Then you have the basement membrane, which is made out of elastic connective tissues, basement membrane, and then you have the endothelial cells of the capillaries. Now there should be two basement membranes because the lining wall of the alveolus and also the lining wall of the capillary. So there should be two basement membranes, but these two bind together, connect together and form a single basement membrane. So you have one side, the pneumocyte type 1, the basement membrane, which is a combination, of the endothelial basement membrane and the alveolar wall basement membrane, and finally you have the endothelial cell of the capillary. So those are the three components of the alveolar septum, that is the barrier between the blood and the air. Okay, so here you can see a diagram. You have the type 1 pneumocyte, the combined basement membrane, and the capillary endothelial cells, right? So the diffusion of air can happen across this membrane according to whatever gas it is on either side. So oxygen will go through the type 1 pneumocyte basement membrane through the endothelial cell cytoplasm into the blood, and carbon dioxide will pass through the endothelial cells across the basement membrane through the type 1 pneumocyte into the alveolar lumen, right? So like that, air will cross between this alveolar septum. Now you must remember that if this alveolar septum is damaged or thickened due to any reason, then this gas exchange is reduced. Now when you have something like an infection of the lung, there's a lot of mucus secretion, then the mucus can get filled inside the alveoli, okay, and then this alveolar septum becomes thick. So if there's inflammation in the lung, this alveolar septum can become thin, and because of that the gas exchange may be difficult, right? So anyone with an infection of the lung can have difficulty in breathing, okay, and oxygen supply may be reduced. Okay, so here you can see what we call alveolar macrophages. Now we did immune system last week, so here we talked about macrophages, who are cells that can phagocyte particles and also they are antigen presenting cells, right? And they are found everywhere where it is where the body is open to the outside environment. So the respiratory tract is a place where you get all sorts of pathogens and different foreign material coming into the body, right? So you get a lot of macrophages inside the respiratory tract epithelium. Okay, so you see alveolar macrophages. Now these alveolar macrophages look black, okay, so they have large nuclei, and inside their cytoplasm you see all these black colored dots. Okay, so these are actually ingested carbon particles. So when you breathe in air which has carbon particles from all the smoke that is everywhere, right? So the smoke carries small carbon particles, and they go and the alveolar macrophages identifies it as a foreign body and engulfs it, and this carbon particles remain within the cytoplasm of the macrophage, because actually the macrophage cannot destroy these carbon particles, so it just holds it inside its cytoplasm and keeps it. And when that macrophage dies, it divides into two and passes on the carbon particles to the other macrophages, right? So so like that, generations of macrophages will be carrying these carbon particles, and that is what actually gives that dark blue or black color to the lung. So if you take a lung of a newborn, it looks pink, right? But as the person grows up and breathes the city air, actually they lived in somewhere where there's not much carbon particles, then the lung would be that blue, black, right? And also in someone who is a smoker, where lots more carbon particles get ingested by the macrophages, you will get a black lung, more black, right? So smoker will have a darker colored lung than a non-smoker. Okay, so alveolar macrophages are generally found in the alveolar lining cells or in the septa, that is between each of the alveoli, right? Septa means between the walls of the alveoli. Then they phagocytose microorganisms and other foreign bodies, and they can migrate up the airway. Actually they can go into the side of the lumen. The macrophages can stay inside of the lumen, and they can migrate up along the airways until they can get sometimes coughed out as well, right? So if they are left inside the septa, the macrophages which are left inside the septa of the alveoli, they give the black discoloration to the lung tissue, as we discussed. Okay, so alveolar macrophages are important for the innate immunity of any pathogen which is trying to get in through the respiratory tract. Okay, so initially alveolar macrophages will try to stop these organisms from invading the body. Okay, so we'll talk about the clinical correlations of the respiratory tract. One of the most common non-infectious or non-communicable diseases is bronchial asthma, right? Bronchial asthma, which is a disease of the bronchioles of the respiratory tract, right? Bronchial asthma. Then you have what is called tuberculosis, which is an infectious disease which used to be very common all over the world, especially in India and Sri Lanka, but now it's much less. We have very few tuberculosis patients. Initially, of course, tuberculosis was completely eradicated, but then with AIDS coming in, again tuberculosis cases were found. Okay, then we have something called the respiratory distress syndrome or the hyaline membrane disease. That is where surfactant is deficient, right? So in some newborn babies, surfactant may be deficient, and because of this, the lungs don't open out, right? So there's a lot of restriction to the lungs, and it does not open out, and the child finds it difficult to breathe, right? So moment they are born, you can see that they are in distress. Okay, so this is a condition that you might see when you do pediatrics, right? And also tuberculosis actually is infection of the alveolar the bacillus which causes tuberculosis gets engulfed by the alveolar macrophages, right? So the alveolar macrophages find it difficult to destroy this bacillus, so sometimes they tend to keep it, just like those carbon particles, they keep it inside their cytoplasm. Alright, so in some people they will destroy it, immunity will destroy it, but in others it might lie for a long time, and when your immunity drops you can get tuberculosis disease, right? As you know, you are given the vaccine called BCG when you are small, and this will, as actually the second day of birth you are given the vaccine BCG, and that will give you immunity against tuberculosis, right? But this immunity is not lifelong, so as you grow older you may be exposed to this disease, you can get tuberculosis. Okay, so this is a picture which shows you asthmatic bronchiole, right? So the normal bronchiole is given, so it has a particular thickness to its wall. It has the bronchial wall, smooth muscle wall, but in asthma the smooth muscle wall is thickened, right? And it is more reactive to different substances in the air. So generally asthma can occur due to cold air blowing in, or due to dust, or due to some sort of infection. Anything with reacts with the bronchial wall will cause these bronchi to constrict, right? So constriction of these bronchioles causes inhaled air to get trapped inside the lung. Okay, so the patient finds it difficult to exhale the air outside. So this reduces the efficiency of the lung, causing distress to the patient, increased rate of respiration, and difficulty in breathing, right? So asthma has to be treated properly with what are called bronchodilators, right? Those are drugs which act on the smooth muscle of the bronchioles and make them relax, so that this restrictiveness or the restriction of airflow into the lung is reduced. Okay. Then here you can see a radiograph of the lung which shows these opacities, the white colored areas in the lung field, which are actually tuberculosis. This is a typical picture of tuberculosis, and in persons who have chronic cough, fever, sweating spells, you have to take a chest x-ray and see whether there are any patches. Okay, and you can also take a sputum sample and examine it, stain it and look at it under the microscope to look at the tuberculous bacillus. Okay. Okay, so this is actually a lung with pneumonia. So you must be hearing a lot about pneumonia these days. So here you saw the normal lung which has small thin walls and the inside is full of air. Now here you can see the inside is full of lymphocytes, right? So lymphocytes, dead and dying lymphocytes, lots of mucus, all that is completely filling the alveoli, right? So here you can see the alveolar septum or the barrier towards the air and blood, between the blood and the air is thickened, completely thickened, and hardly any air can get into this particular lung, right? So that is why people with pneumonia becomes find it difficult to breathe, and they become they need to be ventilated or given oxygen so that their bodies can function. Okay, otherwise there's no oxygen going into the blood when the alveoli completely filled with a pus. It's like pus, right? Sputum, or what you cough out. If you have a infection of the lung, you must have, I mean anyone would have had one. So when you cough out, you get what you call phlegm, right? In Sinhalese you call it sema. Okay, so that is actually lymphocytes and secretions and dead and dying cells of the respiratory epithelium, all those things. So those things which are inside the lung will prevent you from exchanging gas exchange in that particular alveoli, right? So the simple form is what we call bronchitis, that is the inflammation of the bronchial wall, which is a very common condition, and in a few days it will regress and you will be normal, right? However, if you have infection of the lungs which is more severe, when you get, then you call it pneumonia. Okay, so pneumonia may be all over the lung, maybe in part of the lobes, you get what is called lobar pneumonia. So like that, so that part of that lung, though it is getting perfused with blood, is not actually getting aerated. There is no air coming into that part of the lung, into those alveoli. So because of that, oxygenation of the blood which is getting pumped into that area is not happening. So there's a mismatch between perfusion and aeration. Okay, so these are all things to make you more interested and to let you know why you are actually learning these things, right? So these are not really histology or anatomy, but for your interest. Right, so I've just put out some questions for you. These two questions. Describe how the upper airway conditions inhaled air. Okay, that is the first question. And the second question is, list the tissue layers in the trachea and describe how each layer is adapted to perform its function. Okay, so I expect you to write down the answers to these two questions, and you can give me a summary of the answers either on the Facebook page or in your WhatsApp group, right? So I will give you a few days, and then you can post the answers you come up with, and we will discuss that on these uh, two pages, right? Either WhatsApp or Facebook. Okay, so then yeah, so this is actually, can you identify what this is, right? So these three pictures also, I want you to write the answers. Just tell me what this is, right? What the structure is. So the first one, all right, so this is the second one. You need to identify this structure as well, and also give me the reasons for identification, right? So the first one, tell me what it is, what the structure is, and the reasons why you say it is that particular structure. For this one also do the same. Right, so this is the third one. Again, I want to know what this is and the reasons why you identify it like that, right? So this is similar to the OSPE examinations that you will get. Okay, so I expect the answers to the two essay questions as well as to these three OSPEs. Okay, so you need to either discuss it on WhatsApp or Facebook.